Welcome to The Explainer. Today, we're basically going on a microscopic treasure hunt. We're diving deep into our own DNA to figure out how scientists find these tiny but absolutely critical clues hidden inside our massive genetic code.
So let me ask you this. Imagine you have a book, a really, really big book. It's 3 billion letters long, and your job is to find one single typo somewhere in there. I mean, where would you even start? This sounds impossible, right? Well, that's the exact challenge geneticists are up against every single day. And no, that's not an exaggeration. The human genome, our complete set of DNA, has roughly 3 billion base pairs. Those are the letters of our genetic story. Trying to find one tiny variation in a sequence that long is just a monumental task. And that's exactly what we're going to unpack today.
To even get our heads around this, it helps to stop thinking of it as just one long book. Instead, picture a massive, sprawling library. And just like any good library, if you want to find something fast, you need to know how it's organized and, more importantly, what you're actually looking for.
So what are these genetic typos that scientists are hunting for? Well, they're not just random mistakes. They're actually the source of all genetic diversity. And yeah, sometimes they're the cause of disease. Let's take a look at the main things they're searching for. First up, you've got DNA mutations. These are pretty much what they sound like. Sudden, random changes in the DNA sequence. Think of it like a letter getting swapped out, deleted, or a new one inserted. A C suddenly becomes a T. It seems so small, but that one tiny change can have huge consequences for how our bodies work.
And then there's this other thing called gene duplication. This is less like a typo and more like a biological copy-paste error, where a whole chunk of DNA gets duplicated. Now, what's really wild about this is that it's a major engine of evolution. It creates this raw genetic material for new genes and new functions to pop up over millions of years.
Okay, so to keep track of all these relationships between genes, scientists use a few key terms. Homolog is kind of the family surname for any genes that share a common ancestor. Then you've got orthologs. Think of them as cousins. It's the same gene, but in different species, like the insulin gene in humans and mice. And paralogs are more like siblings. They come from one of those duplication events inside the very same genome. Understanding this family tree is totally crucial for tracing our evolutionary past.
All right, so we know what we're looking for. But how in the world do you actually search that 3 billion letter library? You're not going to read the whole thing every single time, right? Of course not. You use an index. And in the world of computational biology, one of the basic indexing tools is something called a keyword tree.
And honestly, it works just like the index at the back of a textbook. You want to find the page about mitochondria? You don't scan every page, you just flip to the index, and it tells you exactly where to go. A genetic index does the same exact thing, just for a computer. It lets it pinpoint a specific DNA sequence almost instantly.
The idea behind it is actually pretty simple. You build this tree-like structure, and every path you take from the starting point, the root, down to the end of a branch, spells out a unique DNA sequence. It's a super efficient way for a computer to check if it's ever seen a particular sequence before, as you'll see right here. For example, let's say we indexed these words. Apple and apropos would start down the same path, first A, then P, before they split off. Same thing for banana and banana. You can see how this structure is way more efficient than just a long list. But for a 3 billion letter genome, we're going to need something even more powerful.
And that brings us to the star of our show, the Burrows-Wheeler transform, or BWT for short. Now, this is a truly brilliant algorithm. It takes a piece of text and, well, it shuffles it. But it shuffles it in this very specific, almost magical way that makes it unbelievably fast to search.
So here's how this little magic trick works, in just 3 steps. First, you add a special character, usually a $sign, to mark the very end of your sequence. Second, you create every possible rotation of that sequence. And third, you just sort all of those rotations alphabetically. I know, it sounds a little weird, but stick with me here. Let's see it in action. Let's take the sequence ABABA. First, we add the $sign. So it becomes ABABA. Then we just keep rotating it. You take the first letter, move it to the end, and do it again and again until you have a full list of every single possible rotation. And now, here it is, the aha moment.
We take all those rotations and we just sort them alphabetically. You see how the first column is now perfectly sorted? The $sign comes first, then all the lines that start with A, then the lines that start with B. But the real magic is hiding in that last column.
You see that last column of jumbled up letters? That is the Burrows-Wheeler transform, and it has this incredible property. Characters that were next to each other in the original sequence now tend to get clumped together in this new version. And that reordering makes the data way easier to compress, and most importantly, allows you to search it at lightning speed.
Okay, so we've got this amazing computer science trick. That's cool, but how does it help us crack the genetic code? Well, this is where the algorithm jumps out of the textbook and into the real world of biology. The main way it's used is for something called read mapping. See, our gene sequencing machines, they don't read the whole 3 billion letter genome in one go. Instead, they spit out millions of tiny fragments called reads. The scientist's job is to figure out where each of these tiny little puzzle pieces belongs on the master reference genome.
And that's the final connection. The Burrows-Wheeler transform is the powerful engine under the hood that lets computers take millions of those short reads and map them back to that 3 billion letter library, and do it in just minutes or hours instead of lifetimes.
So why does any of this matter? What's the big deal? Well, because this incredible search speed allows scientists to quickly find mutations linked to diseases. It helps us compare our DNA to other species to understand evolution. It's paving the way for personalized medicine that's tailored to your specific genetic code. It even lets us build the genomes of new species from scratch. It is, no joke, foundational to almost all of modern biology. You know, it all started with a simple, almost silly question. How do you find a typo in a giant book? And the answer involved this clever way of just shuffling and sorting letters. It really makes you wonder, what other massive, complex codes out there in nature, hidden in data, maybe even in the universe, are just sitting there, waiting for the right clever algorithm to finally unlock their secrets?